to the Weekly Bioanalysis, a KCAS podcast.
Hello again, and welcome to the third episode of the Weekly Bioanalysis. My name is John Perkins, and I'm the Senior Scientific Advisor for all things related to LCMS technologies at KCAS. I've been with the company around 6 months, but have a wealth of experience in bioanalysis for the last 25 years. I'm here with my co-host, Dominic Guarino.
This is Dominic Guarino, the Senior Scientific Advisor at KCAS with a focus on LBA technologies and biomarkers. Like John, I too have about 25 years of bioanalytical experience.
For the first time, if you're watching on YouTube, you should be able to see our contact details as we alluded to it right at the end last week. Today's podcast, as always, is brought to you by KCS, Kansas City Analytical Services. KCS is a bioanalytical laboratory located in Kansas City, Kansas, serving the pharmaceutical and biotech industries for over 40 years now. Dominic and I are senior scientific advisors at KCS, and either or both of us are available to answer any questions you may have regarding this podcast or any KCS services. We're really happy to have you listening again. As mentioned, this will be the third week recording the podcast. So if you missed this last week or the one before that, they're all online. I have to say, this has been great so far. I spend a lot of time online because I'm at home, and there's obviously a lot to discuss in terms of COVID-19 and the effect that has on our business, the effect it has on bioanalysis. So it's great to talk about this on a regular basis. What's your thoughts on this, Dom?
Yeah, it's been really fun the last few weeks. Pleasantly surprised at the interest level in our podcast, and find myself spending most of the week almost obsessing about what we were going to talk about or things to talk about. But yeah, this has been incredibly enjoyable. Really enjoy working with you, John, and looking forward to some continued success.
What has prompted this podcast is the fact that business has changed with COVID-19, where we've had social distancing implemented, and we really want to still be able to contact people and talk to them. So we're using any facility we can find to keep in contact with people. Even today, I'm in upstate New York, and Dominic is in Kansas City. Our production team are also scattered. So this is really one mechanism whereby we can keep in touch with people and keep news about KCS and what we're doing out there. Then, after feedback, we will wrap up and give a little teaser for next week. So again, we're happy to have you here, thrilled indeed, and we're looking forward to a fun episode today. Dom is going to go over today's podcast topic a bit before we jump into the news and resources.
So today we'll be focusing on antibody testing, specifically antibody testing for COVID. Is antibody testing for everybody really feasible? And then we'll be discussing items such as what will the post-COVID world look like? And then we'll even go into a little bit around sensitivity and specificity of some of the tests that are considered good and what that really means. So I think we're going to start with the news. What a jam-packed week it's been, John. I think the first thing we need to go right into is remdesivir. I don't know if I said that right, John. That's more in Joe's house, but that's the Gilead drug that caught major attention this week. There's a study out there now that demonstrated, I believe, about a 5% improvement when you look at placebo groups having a 13% death rate, and then those that are on the drug at like 8% mortality rate. This is considered significant. I think it has some improvements, but certainly this feels like it's jumping the gun early again. What are your thoughts, John?
Yeah, so there's talk with remdesivir that it may well go forward for emergency use authorization fairly quickly. Europe is looking at trials rather than jumping on that kind of process. But equally, when we hear one trial, the NIAID trial looked favourable, there was also a placebo-controlled drug trial in China where the results weren't quite so favourable, but there was some concerns about how that trial was set up. Also, it was looking at severe patients late on, so that might be a factor in where remdesivir might be applicable. It's the first step forward to being hopeful that we'll have something, but there's still a long way to go, in my opinion.
I completely agree, John. It's getting the hype. I hope it works well. I hope it has benefit, but hope is not a strategy, as we all know. And I think part of the challenge we're having right now in the scientific community and throughout the world is people are demanding answers. And the reality is science takes time. And so this continues with the yo-yoing, and it's getting fast tracked, and the president's demanding the FDA approve it. These are Those are things that we don't like hearing as scientists around demanding things, right? So we want the data to tell us what it is. And you mentioned that it was a National Institute of Allergy and Infectious Disease study. Those can be narrow in scope sometimes, and maybe not across as broad a reach as this might need to be. So I think it's to be determined, although let's end on a positive note and say there is something that I hope patients can kind of rest, or at least have some benefit from.
It's a glimmer of hope. I think I've read a number of people saying it's the first step. At least it proves that something can be effective to some extent. So hopefully, like I say, there's hope for other medications in the future. I'm going to grab the next topic actually, where we talk about vaccines now. There has been a lot of discussion in the UK about the applicability of a vaccine developed at the Jenner Institute in Oxford. And that has they've obviously been talking to pharmaceutical companies about partnering, and they've decided to work with AstraZeneca, which has given them access to the capability of scaling up should the vaccine prove effective. So there's obviously a lot of attention to that. It's got that ability to move out the academic realm into the pharmaceutical realm if again it proves effective, but we still have that time factor to consider here. So what's your take on that piece of news?
Similarly, I'm excited about the vaccine race, but caution how fast these things are going to come to fruition, or at least broad coverage. But again, it seems to be somewhat of a nice breakthrough, and I'm cautiously optimistic about it and hope it can continue. But the idea that it's going to be ready in 18 to 24 months.
So on the timing, you're going up against Bill Gates, who says we can do it in 9.
Yeah, I mean, look, I don't tell Bill Gates how to get his word system 30 or whatever we're on out into the market. So I think he should stick to what he knows, but hope we can deliver. But I mean, the reality is there's a lot of, as I mentioned in the previous podcast, you got to take into consideration ethnicities and races and ages and all that sort of stuff. Now, do I think it can be something that can be administered to in a broad reaching clinical trial to numerous people? Sure. But let's not call it an approved vaccine at that stage. Kind of different things there. So maybe we'll move on to the next news and notes.
I think so. Yeah, let's take a break from COVID-19. The pharma industry isn't just focused on COVID-19. There's obviously you've got many companies looking at many drugs for many different applications. And April was interesting in that we actually had 9 drug approvals in April. So we thought we'd discuss the approvals from the previous month. And 9 is a big number. I mean, there's been recent years where we've had 18 over the course of the year. But when you're talking 9 in a month, that's pretty impressive. Breaking them down, it's one product for treatment and control of bleeding episodes for individuals with haemophilia A and B, and there's 6 small molecules, 1 vaccine, and 1 ADC. So it looks like the death of small molecules as effective medications is somewhat exaggerated. Of the small molecules, of the 6, 2 of them are repurposed old medications, reformulations of mitomycin and ephedrine sulfate. But some of the other small molecules are pretty interesting. There's if, there's an insight drug which is there for the treatment of cancer of the bile duct. Neurocrine Biosciences have a drug which has been approved to help extend the effectiveness of levodopa, effectively to extend the episodes whereby it controls the spasms and shakes with Parkinsonism. Seattle Genetics, which I always think of as a large molecule company, actually has a small molecule drug approved for HER2 positive breast cancer, and they end up with a small molecule because of various purchases. The drug originated with Array Biopharma and ended up with Seattle Genetics, and then moving through to approval. AstraZeneca and Merck had a drug approved for symptomatic inoperable plexiform neurofibromas. It's for pediatric patients, so again, it's an indication of how many different indications people are working on. And then for us as bioanalysts, the one that is probably the most interesting is an immunomedics drug. It's actually an ADC where we can sit here. I'm a small molecule bioanalyst. Dom is a large molecule bioanalyst, and we fight over which parts of the molecule we could provide the analysis for. So, what's your take on all this, Dom?
Yeah, John, I think there's a couple takes. One, the death of small molecules is way over exaggerated and overblown. I think there will continue to be this repurposing of small molecules. And then that leads nicely into the ADC space, as well as even Seattle Genetics having approval of a small molecule, even though they're a large molecule. So it's the combination of the two that I think you'll see more and more approvals on. Just because, conceptually, one of the challenges with small molecules has always been it kind of systemically treats. And one of the advantages of a biologic is it's more targeted. Well, I mean, in theory, putting the two together, you've got a real nice solution for cancer. Of course, there's a number of bioanalytical challenges, as we know, John. In vivo, getting the biologic to the point of where it needs to do its action is still somewhat challenging. And then getting the small molecule to be deposited at the right concentration and to maybe either come off the protein or come off the antibody, or how all of that can be challenging. So these are very promising drugs, and I'm looking forward to continuing to help and support drug development in the ADC space. And then I'll just share that we did see one vaccine on there, but I think that's an area where obviously we're going to see maybe potentially a lot more of with the whole COVID thing, right?
Yeah, I didn't mean to brush over the vaccine. I mean, it's there for invasive meningococcal disease. So, again, it's going to be something that hopefully will be of huge benefit to people.
John, vaccines, there are so many out there that would be approved or potentially utilized that are not on the market just due to some of the patients that they are designed to treat. For instance, pregnant women, there's a Gram B vaccine out there, but you tried doing a clinical trial on pregnant women, right? Similarly, there are vaccines in children that take years and years. And I touched on the rotavirus one. That took a lot of activation energy to get that approved. Just because, as you're sitting here, John, would you have let your children be participating in a trial?
Yeah, that's a very tough question, definitely. Yes.
Just things to think about when it comes to vaccines. Well, today we're going to really dive into antibody testing. So, what is antibody testing? So, maybe I'll take a small moment to share with you that within all of us, within our circulatory system, we have things called antibodies. Most people probably have heard the word, or it's becoming much more mainstream. But an antibody binds to an antigen. And so your body has a repertoire of antibodies that your immune system can produce. And that's where a lot of the COVID vaccines are now starting to target getting your innate immune system to prime your adaptive immune system so that they can generate T and B cells against COVID to make antibodies against it. It's no different than like a hepatitis vaccine or other vaccines that are out there. But the antibody testing themselves are typically done in a ligand binding assay mode where you have some sort of antigen that the antibody binds to. And then so you add the patient sample and the antibody binds to it, and then you can detect it. Or you have an antibody against it so that you put the antibody down on the plate, add the patient sample, the antibody binds to it, and then you can come back and do a color change. A lot of the antibody testing we're trying to do today is in the lateral flow, meaning you just pick your finger, put it on a sheet, and it gives you a color change, similar to a pregnancy test. That works real well in a pregnancy setting. However, antibodies are very tricky little beasts.
Tricky big beast, given the molecular weights that you're talking. These are hefty things to deal with.
Absolutely. And certainly a lot of false readings. One must think about that. I mean, I think we sort of talked a little bit about that, and maybe it's a nice segue into what we're seeing in terms of antibody testing for COVID. There are four, I think there are two more that were approved within the last week. I think there are six recognized antibody tests. And then there are about 125 on the market that are variable between 70 to 80% false rates. I mean, John, this is kind of scary when one thinks about what one's samples are being done with, what people are doing to measure COVID antibodies.
So locally, we actually have a company are combining with our local hospital, and they're looking to provide antibody testing for all their essential workers. So it's a locally developed antibody test that they're looking to make available for all essential workers. So I'm not sure what has happened for them in terms of approval, but it's a measure of what kind of effort is being placed on getting testing in place for people to help us get back to a normal way of life.
Yeah, of course. There's similarly here in Kansas, a site that I used to work. They're starting to reach out into the community and have antibody testing readily available. Is that really a good thing, you think? Meaning, pop-up antibody testing kind of scares me a little bit.
I agree. I'd much rather see all this stuff coordinated on a national level. I don't. I really, yeah, this sort of piecemeal approach to everything. Actually, to be honest, not just on a national level, on an international level. I mean, I've been thinking about it, and it's baffling to me when you've got a pandemic how little international cooperation there seems to be on this so far. It's sort of each country's taking its own approach, and in this country, it's broken down even further, and that seems counterintuitive to me. I would think you'd want to pull all resources and look at what's worked, what hasn't worked, and how we're all in this together, and how do we fight it together.
Yeah, I would agree. Typically, when it comes to the vaccine space, it seems like the communities are coming together. When it comes to the antibody, it's pretty polarizing. It seems like it's a race to the market, right? So, John, to your point, there are only a limited number of labs that currently have antibody testing available. And when you talk about the international community here at KCAS, we're actually in the works of working with an international company to not just evaluate their COVID antibody test. As you know, we do a lot of anti-drug antibody testing, which is a much more sophisticated and a much more rigorous validation process than what's available for antibody testing for COVID-19, for example. We do much, much more in the realm of evaluating the drug's impact on the production of antibody, meaning we have multiple tears, we have to screen, confirm, and titer, and then go to neutralizing antibody in the anti-drug antibody space. With that said, we're in the works of partnering with an antibody production company. They make antibody ELISA kits. They've been making them for a long, long time. And we're in the works of having that be evaluated at a laboratory and potentially validated. And we're might be one of those individuals that throws our hat into the ring offering antibody tests for COVID.
As long as it's validated, I'm happy to stand behind that.
Like I said, we're going to definitely take an approach that's much more rigorous than, say, a lateral flow. This will be a full 6 or 8 hour ELISA that will require a pretty sophisticated validation and evaluation. But we have worked with this company in the past, and their ELISA kits are pretty rock solid. So I'm pretty optimistic about it. Do we think there's going to be antibody testing for everybody in the world? Do you see a day where you're walking into the movie house and you got to put your finger on something to give a drop of blood that tells you if you're infected or have antibodies? Or
I hope we get to vaccine before that. I haven't really given that too much thought, but I mean, again, it's we've got that balance between we have this virulent virus to deal with. We're trying to get back to economic normal. What is our pathway to doing that? I think it is going to be a case of how do we ensure people are protected if we're out working and mixing as we used to. So I honestly, I don't know quite what it will look like, but that maybe we'll go in that direction.
Well, I mean, I'll give you an example. NBA teams are trying to procure 15,000 antibody tests so that they can start testing people who come in the arena or want to test all of their players. And there's talks of them resuming like down in Florida and playing in Orlando, just on the Disney property because it's all sitting idly right now. And they've got lots of gyms down there apparently that they can play in. This is stuff that might happen this summer. I know you were like, "Oh, I hope we get a vaccine," and I completely agree. But I think you're going to see a lot of economic impact, meaning people are going to, procure all these antibody tests. They don't really work that well. That's and go ahead and finish your thought, but I'll share with you a little bit of some stats around how these things perform.
Yeah, so and there you are. If it's not working well, you've just then crowded a bunch of people in an environment with a disease that spreads fairly easily, which is counter to what we're all doing at the moment. So
I don't even know the purpose of it, too. I mean, there's talks of it, but just because you have the antibody doesn't mean you're not susceptible to getting infected again. There's a big misnomer, right?
yeah, there's still that concern. I mean, I've seen a number of articles that said having had it once is no proof against having it a second time. And yes, I don't know what it necessarily proves at the moment either.
Yeah, so that's the skinny on all of this. If you test positive, is this going to give some individual a false sense of like, gee, I don't have to observe social distancing. Gee, I'm fine. I'm protected the rest of my life. Those are the types of things that we certainly don't want science to give people like, a false sense of being Superman, so to speak. Boy, I have so much fun making this podcast. I find myself looking at MIT videos. I'm just so impressed that I watched something from MIT and kind of understood it, right? But what this video broke down was, you hear the term specificity and sensitivity of a method. And if I said it had 95% sensitivity and 90% specificity, John, that sounds pretty good. For an LBA method, that's really good. But this individual showed that in COVID IgG testing, the predictive value changes significantly, meaning if the prevalence is 5%, the predicted positive value is only 34%, and the negative predictive value is 99.8%. What that means is 34% of the time, it's missing. If the prevalence is 30%, the positive predictive value is 80%, and the negative predictive value is 97.7%. So that to me really means we have a lot of work to do to figure out what the prevalence is, and then we have a lot of work to do to figure out what our IgGs, that's what this is in particular, COVID IgG test is doing.
It all points to the fact that we've got so much to learn about this to get even through to the other side.
There are a lot of things being lifted, and even as we were getting prepared for this podcast, breaking news says that we have a couple scenarios that may come in the next 18 to 24 months, and these are some of our leading experts in this space are saying, hey, this thing's not going away. It's going to take 18 to 24 months for herd immunity to take effect. And so with lifting of these restrictions, I believe we are going to see somewhat of a spike. I mean, the expectation would be hopefully it's not like driving us right back into where we are today, although the experts feel like that's kind of the scenario they're painting. I mean, John, I don't know if you had a chance to dive into that article, but what they're kind of saying is there's one of two or three scenarios, and they really feel like the scenario we should brace ourselves for is some dispute here, but we're going to be right back in this in the fall, and we should be prepared to be rehashing what has occurred over the last 3 months.
Yeah, I haven't seen the article in detail, but I tend to take the cautious approach on this too. And certainly I've seen, a number of graphics based on the Spanish flu from 1918, where, places had parades. Philadelphia had a parade and then saw a massive uptick in cases with that, where they thought they were in control of it. So, you've got to be really cautious with this, because what we don't want to be doing is thinking, yes, we've got it under control, and then consign ourselves to several more months of being at home and not being able to do what we really want and need to do.
Switching gears, John, got something random for you. Do you know what receptor the coronavirus attaches to in the human body? This came out this week.
ACE2, isn't it?
Oh, man, you caught me on that one. You are correct. It's the angiotensin converting enzyme 2. That is exactly what it binds to. That was something that came out over the internet this week. Obviously, you must have seen it.
Hell yeah, I spend way too much time online looking at this stuff.
It's impressive. You're a small molecule guy diving into all this receptor stuff.
There's also the concern about small molecules, and their effect on ACE2 in terms of boosting it. I think it's like blood pressure medication and other things like that has certainly been out there. So I don't know what the latest is on that in terms of any conclusions, but there's certainly that concern has been raised.
Another kind of interesting little tidbit that I came about is something called CTAP. This is the Coronavirus Treatment Acceleration Program. This is what the FDA is putting out there for all those people that are trying to get into the coronavirus treatment. I don't know if you were aware of that, John, but this CTAP is now available for companies, so pretty cool. One last thing here, John, is this Anovo Pharma story. Have you heard a little bit about this one?
I found out about that a little bit before this podcast, and I'm not going to steal your thunder because I think you're the one that passed it on, so you're the one who should talk about it.
Well, for those that don't know, there's a company called Theranos, and they've been accused of being Theranos-like. Theranos, big HBO special on it. They're the company that just committed fraud at a level that is unprecedented in the history of science.
What's her name? It's Elizabeth somebody, isn't it?
Correct.
Way back when the share price flew up with Theranos, and I know that Elizabeth Holmes was in the media all over the place, promoting what they were doing. But it all came crashing down very quickly as well as people began to dig behind the science.
Absolutely. She thought of herself as the female version of Steve Jobs. She was very arrogant about it. And even to the last moment, continued to commit fraud, like on an epic level. They were just simply like taking blood tests and from that were being collected at Walgreens and CVS and literally shipping them off to like LabCorp to get the results and then saying their instrument were doing it. Oh, if you haven't seen that series, it's scary. And it probably rocks the moral fiber of all of us that are in science. Well, this Anovo company, they're saying is kind of similar with what they're doing on their vaccine.
Just to clarify, it's a separate research company are saying they don't trust Inovio, so that's where we stand on that. So it's another watch this space. But it certainly, I mean, Inovio have been very high profile in terms of the early days of looking for a vaccine for COVID. So it is definitely going to be interesting to see where this goes and whether Citron Research's accusations are borne out. Obviously, we can't pass judgment on that right now, but we will watch with interest, definitely.
Absolutely. You're so right. We have no stake in this. We have no facts other than what we can read online. But they did get on 60 Minutes, the senior vice president, and sort of start talking about how they got this done within a few hours. More to learn, more to hear about on that one.
I'm really interested, Dom. Have you watched Contagion recently at all?
No, I have not, but I do want to watch that one.
It's the Steven Soderbergh film, and when things started to move, you're really starting to hear more and more news. My wife and I watched it, and it's like, I think probably the first sort of 80%, maybe beyond that, is great. It's done really, really well in terms of ease of transmission and how the scramble to try and address it and just some of the other effects of the virus on people. And then at the tail end, it just sort of seems to wrap up too quickly. It's almost immediately we have a vaccine. And I know you can't say, oh, here the vaccine that they had research of took 4 years to get to this point, but it does sort of brush over that point, which I think sort of lets down the film to a large extent when you've got the background we have. But it is a fascinating watch based on where we are right now. I would totally recommend revisiting it.
It is skyrocketing on Netflix, and that's why it's popped up on my radar. I try to see what the top 10 in Netflix are. I fully expect to catch it this week.
So we're going to go ahead and continue with the feature that we introduced last week with the feedback from fans. And first up is something from Bob. Kudos for KCS thinking outside the box and implementing new ways of reaching people, especially during this time of change. Thanks a lot, Bob. We actually are exploring many other avenues of how we can get the message out there, whether it be informal like this podcast, or another thing that Dom and I are both doing is we have 2 Minute Tuesdays where we talk about a quick snippet about some of our areas of expertise. Those are out on YouTube as well.
The second feedback we got is from Anonymous. It says, I'm trying to get my boss to implement new creative ways instead of just white papers, white papers, white papers. This podcast will do well for you. Wish we could do something like it. Wow, thanks a lot. Anonymous, that's a testament to our executive team for allowing us to put this podcast on. Thank you very much.
Exactly. Yeah. It's great to have the backing to spend time on putting this together. Moving on, Judy says, "Thanks for posting this. I listened to the second episode about vaccines. Info was easy enough for me to understand. The guys were interesting and funny. I'm not sure my wife would agree with that, but I actually do appreciate the feedback. Thank you very much. I'll hold that over her."
I don't know about us being interesting, John, but certainly appreciate that sort of feedback. The next feedback we received is from Erica. Thanks for sharing. Very much enjoy the podcast so far, guys. Well, thank you, Erica. We hope this third one doesn't let you down.
Next up is Mary. The episode about vaccines offered answers to questions that so many people have. I hope this episode makes the rounds so more people can hear these simple to understand explanations about vaccines and treatments for COVID-19. We hope so too. I mean, from our perspective, we're very involved in the bioanalytical for COVID-19, where we obviously have to stay on top of what's happening out there, but everyone has questions, and if we can use this mechanism of simplifying it so people understand what's going on, then we'll do more and more of these, because I think it's absolutely vital.
John, you heard me mention we may have an antibody test coming out there. Got news this morning. It looks like the first samples from a COVID trial for compassionate use of a small molecule will be coming to KCAS within the next week or two. We'll just continue to do whatever we can do to help support the COVID-19 initiatives, whether it's through antibody testing, support of drugs, or even vaccines.
That's great news. Excellent news.
John, got something for you. Guess what I was able to find in the grocery store?
Toilet paper?
No, whole dry milk. Guess what we're bringing back tonight? It's Pizza Friday, baby. Gonna make myself some dough. Gonna get my pizza going. I can't believe it. I was so pleased when I found that.
I thought you were going to use it as a surrogate matrix for like optical drug analysis, because that's what we've used in the past.
You're absolutely right. It does make a good blocker in LBA assays, but I couldn't believe it. I'm going to be firing up my pizza stone tonight, and we're going to get our pizza on.
Yeah, I'm just counting down to the day where I actually can find Marmite crisps on britishcornershop.co.uk. We have regular orders there, and that's the one thing I crave.
Yeah, John, I don't even know what you said. What is a crisp?
It's potato chip, but we call them crisps in the UK, and Marmite is the spread that is a byproduct of brewing, and it's a real love it hate it thing. But my wife loves Marmite, but she hates the crisps. I love the crisps, and the spread I'll happily have as well. But yeah, it's a British thing.
Oh, well, I think I'd be more like you and enjoy the crisps. John, so far, the podcast has revolved around COVID. Let's face it, it started during COVID, so it made sense to kind of have it focused on COVID. Well, next week, we're going to talk about all things unrelated to COVID that are going on in drug development and drug approval.
Yeah, I think that's a great idea. I mean, as we've talked, the drug development process takes a lot of time. So if we just focus on COVID, then, we'll be talking the same thing week in, week out. We'd rather build on what we talked about this week. That we looked at the drug approvals in April. And so then it makes sense to have a wider view at what's going on in terms of the pharmaceutical industry and news there, and bioanalytical as well. And I welcome the change, and then we can catch up on COVID in a couple of weeks' time, because I'm sure things will have happened between now and then, not least updates on Inovio. As always, if you have any questions about the podcast, let us know. As I mentioned earlier, you'll see our contact info at the bottom of the screen, and we can also be reached at kcsbio.com. We'd love to hear from you. Send us your thoughts, your questions, your feedback, any comments on bioanalysis, any comments on the podcast, or just let us know what items you've recently seen on your grocery shelves that have returned at last.